Welcome to this deep dive into pharmacodynamics. At its simplest, pharmacodynamics is the study of what a drug does to the body. It explores the biological effects produced by a drug, the site where it acts, and the mechanism by which these effects are achieved. It is important to distinguish between drug action and drug effects. Drug action is the initial response produced when a drug reaches its site of action, for example, a drug stimulating a muscle. The drug effect is the resulting physiological change, such as the contraction of that muscle. To elicit an action, most drugs must bind to a receptor. Receptors are specific binding sites, usually proteins, located on the cell membrane or inside the cell. Think of the receptor as a lock and the drug as a key. For a drug to work, its molecular shape must be complementary to the receptor's binding site. When they bind, they form a drug-receptor complex, which then triggers a biological response. But not all drugs interact with receptors in the same way. We define this interaction through two key terms: affinity and efficacy. Affinity is the ability of a drug to bind to the receptor. Efficacy, or intrinsic activity, is the ability of that drug, once bound, to actually activate the receptor and produce a response. This brings us to agonists and antagonists. A full agonist has both high affinity and high intrinsic activity, producing a maximum biological response, much like the body's own natural chemicals. A partial agonist, however, may bind well but has lower intrinsic activity, meaning even at high concentrations, it cannot produce a maximal response. Then we have inverse agonists. These are fascinating because they don't just block a receptor, they produce a response that is exactly opposite to that of an agonist. Finally, there are antagonists. Antagonists have affinity. They can bind to the receptor, but they have zero intrinsic activity. They essentially act as a blocker, sitting in the lock so the real key cannot enter, thereby preventing any biological response from occurring.
Now let's look at how we measure this response. In 1937, Clark proposed the rate and occupancy theory. He suggested that the biological response is directly proportional to the number of receptors occupied by the drug. He initially proposed an all or none phenomenon, where a receptor is either fully activated or not activated at all. However, we later discovered that this wasn't entirely true, leading to the discovery of partial agonists and the concept of spare receptors. Spare receptors are receptors that remain unoccupied even when the drug has already produced a maximum response. This means the body is highly sensitive and can achieve a full effect without needing every single receptor to be filled. Moving into the specific mechanisms, let's discuss type 1 receptors, the channel-linked or ionotropic receptors. These are essentially gates. These receptors are ligand-gated ion channels located on the cell membrane. When an agonist binds to the receptor, the channel physically opens. This allows specific ions, like sodium, potassium, or calcium, to flow into or out of the cell. This movement of ions changes the electrical charge of the cell membrane, leading to either depolarization or hyperpolarization. For example, GABA receptors and nicotinic receptors work this way. When an agonist binds, the gate opens, ions rush in, and a rapid electrical signal is triggered. Antagonists, on the other hand, block these gates, preventing ion flow and stopping the signal. Local anesthetics are a great example, as they block sodium channels to prevent pain signals from reaching the brain.
Now we move to the most diverse and complex group, type 2 receptors, known as G-protein coupled receptors, or GPCRs. Unlike ion channels, these don't open a gate. Instead, they act as a relay system, using a protein called a G-protein. A G-protein is a trimer, consisting of 3 subunits, alpha, beta, and gamma. In its resting state, the alpha subunit is bound to a molecule called GDP. When an agonist binds to the GPCR, the receptor changes shape and activates the G protein, causing the GDP to be replaced by GTP. This triggers the alpha subunit to break away from the beta-gamma dimer. This active alpha GTP complex then travels along the membrane to find an effector, usually an enzyme. One of the most important effector systems is the adenylate cyclase system. If the G protein is of the G-stimulatory type, it activates adenylate cyclase, which converts ATP into cyclic AMP, or CAMP. This CAMP acts as a second messenger, spreading the signal throughout the cell to activate protein kinases, which then change the cell's behavior, such as increasing the force of heart muscle contraction.
Conversely, G-inhibitory proteins can shut down adenylate cyclase, decreasing CAMP levels and suppressing the cellular response.
But GPCRs aren't limited to CAMP. Another critical pathway is the phospholipase C, or PLC system. In this pathway, the activated G protein stimulates the enzyme phospholipase C. This enzyme then cleaves a membrane phospholipid called PIP2 into two powerful second messengers, DAG and IP3. DAG, or diacylglycerol, stays within the membrane and activates protein kinase C, leading to the phosphorylation of various proteins. Meanwhile, IP3, or inositol triphosphate, is water-soluble and diffuses into the cytoplasm. It binds to receptors on the endoplasmic reticulum, triggering the release of stored calcium ions into the cell. These calcium ions then bind to a protein called calmodulin, which activates further enzymes, often leading to muscle contraction or secretion. Finally, some GPCRs skip the second messengers entirely and directly regulate ion channels. For instance, when acetylcholine binds to muscarinic receptors in the heart, the G protein directly opens potassium channels. This allows potassium to leave the cell, causing hyperpolarization and slowing down the heart rate. This direct regulation allows for a very precise and fast control of cellular electrical activity. Next, we have type 3, kinase-linked receptors. These are essentially enzymes themselves. A prime example is the tyrosine kinase receptor. When an agonist, like insulin, binds to the extracellular part of the receptor, it causes two neighboring receptors to come together, a process called dimerization. This brings their intracellular kinase domains close enough to phosphorylate each other. This autophosphorylation creates docking sites for signaling proteins, triggering a cascade of events inside the cell that regulate growth and metabolism. Lastly, we have type 4. Receptors that regulate gene transcription. Unlike the others, these are not on the membrane. Because their agonists are typically lipid-soluble, such as steroid hormones, they can slide right through the cell membrane. Once inside, they bind to receptors in the cytoplasm or the nucleus. The drug receptor complex then binds directly to specific sequences of DNA. This triggers the transcription of DNA into messenger RNA, or mRNA, which then travels to the ribosome to synthesize new proteins. This process is slower than the other mechanisms, but has long-lasting effects on the cell's structure and function. In summary, drugs can act through rapid ion channels, complex G protein relays, enzymatic kinase cascades, or by rewriting the cell's genetic output. Understanding these pathways is the foundation of pharmacology, allowing us to design medicines that precisely target the biological machinery of the human body. Thank you for watching.